Well, as we all know, the world needs more trees. We need more trees both for the solution of global problems and the solution of local problems. And of course, we also need trees for timber and pulp. The problem we have is that just as we need more trees in the world, we're losing trees, partly because of biotic threats that are travelling around the world due to global trade. The map just shows shipping routes in the world. And also, it's harder to grow trees because of climate change, and it's harder to decide which trees to plant where in a changing climate. So what can we do in this situation to have more trees in the world? Well, for millennia, we've needed more wheat in the world, and we've found that there's a lot of things that we can do about that. If you think about the wild progenitors of wheat and their yields, and the yields that we can get today from modern wheat cultivars grown in agricultural conditions, we have massively enhanced the output of wheat plants. By contrast, if we think about our trees, and particularly our broad-leaved trees, we've hardly even begun to do that process. Tree breeding is basically in the Neolithic. The best we have is semi-domesticated trees. We haven't fully domesticated them, and we certainly haven't done breeding programs for our trees to enhance their pest and pathogen resistance and their yield under different environmental conditions. Part of the reason for that, of course, is that trees, and particularly broadleaf trees, are experimentally highly intractable. So I've got a little table here just highlighting differences between wheat and ash trees, a randomly selected broadleaf tree. Wheat has a generation time of less than a year. Ash, it's over 10 years before they start to flower. Time to harvest for wheat is less than a year. Ash, probably over 30 years if you want timber. The space to grow a wheat plant is tiny compared to the space you need to grow an ash tree. Inbred lines, if you want to do modern genetics, they're available for wheat. They're easy to make because of the short generation time. For a tree like ash, they are unavailable and very hard to make. So in all these ways, something like ash is much harder to work on than something like wheat. Until you get to genomics, the genome size of ash is 0.9 gigabase pairs, as opposed to the genome size of wheat, which is 17 gigabase pairs. So to do genomics on ash is going to be a lot easier and cheaper than to do genomics on something like wheat. So maybe this gives us an opportunity in the genomics era to turbocharge our efforts to have trees that are more productive and able to grow in changing environments. So I'm going to outline 3 approaches that my research group has been using over the last few years to try to understand the genomic basis of traits in trees and how they can be locally adapted. So I'll talk about a genome-wide association study, a phylogenomic method that we've developed to try and find candidate genes, and a genome environment association study. And all of these are papers that are currently in submission to journals, and 2 of them are on bioRxiv at the moment, and I'll flag that as I go along. And the first 2 are based on ash trees. The GWAS is about ash dieback, and the phylogenomic study is about emerald ash borer. And I don't need to introduce them because they were introduced to us very well in the conference yesterday. So for our genome-wide association study, we were very fortunate to be able to use a mass screening trial that Nicola Spence mentioned yesterday in her plenary talk. 144,000 ash trees were planted out from 15 different provenances of ash, and you can see those circled in yellow on the map. And they were planted at 14 different sites in the southeast of England where they could be screened for by natural inoculum from ash dieback in the wider environment. And we focused mainly on 2 sites that established very well and then got heavily hit by ash dieback, one in North Birmingham in Norfolk and one in Frant Place down near Tunbridge Wells. We scored the trees in autumn 2016 for their health status. On the horizontal axis you can see the different provenances of the trees, and then you can see the percentage of them up the vertical axis with different health scores. And the green coloration means a healthy tree, Brown, black and grey are different ways of being dead, and then the intermediate colours are intermediate stages of health. So you can see that from all provenances there were some trees that remained healthy under inoculum pressure, but many of them died. So what we did is we went back the next year and we looked again at 38,000 of the trees, and we took out all of the trees that were still completely healthy. We found we could get 625 trees that were still completely healthy out of those 38,000 trees. So for each of those healthy trees, we chose another tree from the same provenance that was almost dead. We'd have liked to have dead trees, but we can't get DNA from dead trees, so we had to have the almost dead ones. So we didn't get the really, really susceptible ones because they'd already died. But we had these pairs of trees, healthy and unhealthy, from the same provenances. And on that we did a GWAS. And this part of the diagram is the GWAS we did. Now, as many of you know, if you work on trees, you can't necessarily get as much funding as people who work on other plants, so we wanted to keep it as cheap as possible. And we did that by pooling our DNA from our different trees. So we pooled our healthy trees from the same provenance and our unhealthy trees from the same provenance in two different pools. So we had pairs of pools, same provenance, healthy trees and unhealthy trees. And so that meant that we could get allele frequencies in those two different pools. So rather than compare individuals, we could compare allele frequencies from populations. And actually, if you do that, you don't lose very much information. You don't really need the information at the individual level. So we did that for many different pools. We put them into a GWAS, and then we looked for genes associated with tree health under ash dieback inoculation. Here's a PCA just summarising the information from each of these pools. And you can see along the bottom, Germany is in black, England is in red, Ireland is in yellow, Scotland in blue, and Wales in green. And you can see that the geography of Europe is roughly reflected in the genetic variation that we found in our pools. You can see the Scottish ones up the top there, German ones down here, and English and Welsh and Irish ones there in the middle. And so that reassured us that we hadn't muddled up our pools and that the provenance data that we had were correct, because we'd expect most of the variation to fit with geography. We then did a genome-wide association study. Here's the Manhattan plot. The green line, which you can probably hardly see at the back, is minus log 10p of 13. And you can see we had many significant hits in our GWAS. 203 SNPs were significant above that level. And several of these hit or were very closely associated with genes that have previously been identified as being involved in disease resistance in things like Arabidopsis. So I'll just give you a couple of examples. This hit here that's shown in blue is an FBOX gene, and these have been many times associated with disease resistance in other plants. And within that gene we had a missense variant, so it actually changes the amino acid in the protein. Here's another example, a CPR5-like protein that modulates salicylic acid, which is involved in response to disease in other organisms. And again, this had a missense variation, a missense mutation within it. So we were quite encouraged from those results that we probably had hit genes that really are involved in resistance to ash dieback. But we wanted to know, could we then take that information and predict the health of ash trees, other ash trees that hadn't been included in our GWAS in the experiment? And so we did genomic prediction. We took 150 trees, half healthy and half unhealthy, and this time we did sequence individual genomes. So we sequenced the whole genome of 150 trees, and then using our SNPs from the GWAS, we tried to predict the health of these trees. We calculated for them a genomic estimated breeding value. And to test the accuracy of our breeding values, we thought, well, what we'll do is, in a breeding program, you would want to pick out the trees that are the most resistant for a breeding program. So we said, okay, if we take the top 20% of our genomically estimated breeding values, how many of those trees really will be trees that are healthy in the trial under ash dieback in real life? And with a training set of 50,000 SNPs, and then just using 200 of them to genotype the trees themselves, with just 200 SNPs, we could get roughly 90% accuracy of assignment to the right health status. So just using that genetic information, we can predict which trees are good for a breeding program. And so that could potentially accelerate breeding programs in the future. Another approach we've been taking to find candidate genes is phylogenomics. And we've done this in the context of emerald ash borer. We had preliminary reports that some ash species from around the world, particularly those that have co-evolved with the emerald ash borer within its native range in Asia, actually have resistance to it. And so we tested this with Jennifer Cook at the USDA Forest Service in Ohio. She collected together living ash species from collections within the USA and then exposed them to the eggs of the emerald ash borer and counted how many larvae actually get killed by the tree. And you can see here that many of the Asian species like Fraxinus manchurica, Baroniana, chinensis actually kill many of the larvae that enter them. Whereas species like Fraxinus americana and Fraxinus pennsylvanica can hardly kill any of the larvae that enter them. So we took this data about resistance in different species, and then for the whole genus, we sequenced the genomes of diploid species. We'd already sequenced the genome of Fraxinus excelsior, and we used that as a reference to annotate the other genomes that we sequenced for Fraxinus. We then built a phylogenetic tree based on our phylogenomic data, and we mapped onto that the susceptibility of the different species of ash. And we found that resistant species, which are shown here in green, actually come out in 3 independent lineages of the ash phylogeny. And so this gives us a chance to look at convergent evolution among these different lineages. If we can find loci that have convergent evolution in these lineages, that could well be something that's involved in resistance to emerald ash borer. And so we did this for 3 different comparisons, and we found a total of 53 genes that had amino acid variants within them that seemed to be convergently evolved in resistant species. We looked at some of the functions of these in model organisms, and 7 of our candidate genes, 7 out of 53, have putative roles related to the phenylpropanoid and flavonoid biosynthesis pathways. And these pathways have often been associated with insect resistance in different species. Here's the KEGG pathway for the phenylpropanoid biosynthesis pathway. And just highlighting here 4 of our genes, you can see that they're heavily involved in fairly central functions within this pathway. And one of them in particular, called CYP98A3, it has 4 amino acid variants within it that we found, and these are all pretty close to the active site of the enzyme. And this is a very crucial bottleneck of the phenylpropanoid pathway, potentially sending things off into different parts of that pathway. This model was built by my PhD student Will Plum, and most of this project was done by my postdoc Laura Kelly. 15 candidate genes had putative roles relating to perception and signaling for defense response. And here's just a list of a few of the Arabidopsis orthologues we found. If any of you are working in this area, some of these genes might pop out to you as interesting. And so again, we were encouraged that this phylogenomic approach could be revealing to us some of the genomic basis of resistance to emerald ash borer, which could inform future breeding programs. Another approach we've been taking is a genome environment association study. And this is to try to estimate local adaptation in natural populations. Now, we did this on dwarf birch. Here's my PhD student, James Burrell, who did the work. We had a debate throughout his PhD whether this was a shrub or a tree. James insisted it was a tree. I thought it was a shrub. I'll call it a tree for the purposes of this talk. Smuggle it in there. This is nationally scarce in the UK, and herbarium records show that just over the last 300 years it's retreated considerably in its range. We sampled 29 populations. That's all the populations that we could find, and we looked at about 15,000 SNPs using RAD sequencing. And we tried to find SNPs where the allele frequency in the different populations correlated with environmental gradients found among those populations. And so we would effectively have an analysis that looked a bit like this. For now, just look at the black lines and black dots. Ignore what's in colour. And each point here is a population. And you can see annual mean temperature in these different populations on the horizontal axis. And then on the vertical axis, you can see a summary of allelic variation at various sites that seem to correlate in their allele frequencies with annual mean temperature. So this is made up of 17 different loci in the case of annual mean temperature for these populations. And you can see this gradient of allele frequencies in these 17 loci. And what we reasoned was, if there's adaptation going on, if you're on the black dotted line, we could predict that you are well adapted. If you're off the line, you might be poorly adapted. And so we can use that to estimate how well adapted different populations are, and then predict how you would need to move them or how you'd need to enrich them with seed from another population in order to bring them to a state of being more locally adapted. And so we came up with a scheme, a hypothetical scheme for how you could move seed around between these different dwarf birch populations to try and promote their local adaptation. And we're now rolling this out on a much bigger scale in a study in Ethiopia on different crops. And James is at last working on something that is almost the size of a tree, although it still isn't actually a tree. So I'd like to thank the various postdocs and PhD students that have been involved in all of this work, and various collaborators and funders. And thank you very much.